From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 47, recorded on September 17th, 2019. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Utah, Nels Elde.
Hey, Vincent. Great to be with you again from Elde Lab Studios.
Hey, we're moving on to our 48th episode, which will be 4 years, right? 4 years of TWiEVO.
That's right. Coming up on our 4th year anniversary. Pretty soon, TWiEVO itself will have a sort of evolutionary record.
That's right. Can we do a phylogenetic tree of TWiEVO?
That's right. We'll sequence some episode genomes and try to reconstruct the history of them.
Yeah.
It's been a fun ride so far.
Can't believe it's 4 years. Huh?
Time flies when you're having podcasting fun.
48 episodes next month. This is October, I guess. Yeah. Well, next month is October, right? The following month, we're going to do something in Europe, you and me and Condit.
Yeah, I'm looking forward to this one. So this is the giant virus meeting happening near Munich, and some sort of hybridization event, an admixture of...
Admixture, good. That's a good word for today.
You'll forgive me previewing some of the scientific energy of TWiV, This Week in Virology with TWiEVO, This Week in Evolution. And I actually can't think of a better topic to kind of mix the two than these giant viruses, which are... these massive, in some cases, virus particles bigger than bacterial cells and genomes as well. And tons of interesting genetic stories and history, just barely tip of the iceberg.
That'll be fun. Mm-hmm. Did you get a list of the people who are going to attend that meeting?
At least a partial list. And definitely, for me, some science heroes kind of in the mix of people whose work I follow and admire since, I have to confess that the viruses who we study are pretty modest in genome size compared to these giant viruses, sort of the title of the meeting. And so I've been following a lot of that work and admiring it since our viruses are pretty small and tame compared to some of these wild beasts that are being pulled out of tundra and water cooler towers and everywhere else. So
I'm at the page for the giant virus meeting.
Mm-hmm.
It says a detailed program will be available once registration is completed, but we can already reveal that Vincent Racaniello and friends will record a podcast for This Week in Virology on Monday night. That's the first night we get there. Holy crap, right?
Oh, I have to be ready. Get our acts together ahead of time.
I'll be tired.
A little jet-lagged podcasting. I'm sure you've done that before.
Yeah, I've done that. I'm looking at the list here. Preliminary speaker list. I got Nels Elde, Rich Condit.
Yeah.
Let's see. Of course, Eugene Koonin will be there.
Yep, that's a good one. He's done a few TWiV episodes.
He has, yeah.
Yep. Mm-hmm.
Now, we'll have to put together we'll have to put our heads together and decide.
Yeah, definitely try to tap some of the speakers on the shoulder or other participants and see if we can get them to sit in with us and share some of their science that they'll be bringing to the table at the meeting.
Yeah, we have to do that. Okay, meanwhile, we have a little paper here. Before we do that, though, everyone who's listening, please consider supporting us financially because we do this on our own finances. Microbe.tv/contribute. Tell you more about that later. Admixture, huh? What's admixture, Nels?
Yeah, so we are, I kind of hinted at that. So admixture, you put in a great definition here, the presence of DNA in an individual from a distantly related population or species. So this is a consequence of interbreeding between populations. I think it's even been referred to as genetic pollution. When you're
That's good.
Yeah.
So that doesn't apply to us and Neanderthals, does it? Because they're not distant enough?
I think that's correct. So I think the distinction you'd make there, and if any of our sort of dyed-in-the-wool genetics listeners or population genetic listeners want to correct it, I think that you'd refer to as introgression.
Yeah.
So you have sort of populations that are interacting anyway, and then exchanging genetic material. Here, for admixture, the idea is that the populations have been removed from each other for longer periods of time. And so another idea is that it's sort of the introduction of foreign or unadapted genotypes. So really coming out of genetic left field, so to speak.
So our ancestral DNA wouldn't count because there's so much of it, right? Like prokaryotic mitochondrial DNA, et cetera. That wouldn't count as admixture. It would have to be more contemporary.
Well, yeah. And I think that's right. So those events also, I would say, aren't kind of the maybe under the auspices of interbreeding. So this is right, like yep. So these are kind of wild, symbiotic events, basically, right? Where an entire genome starts to co-localize in the same cellular compartment as another entire genome, sort of on the origins of mitochondria or other organelles like that. And so there actually isn't genomic and kind of interrelationships going on. There's no recombination. That's what I'm trying to say. At least at the outset, there are kind of two genomes held nearby, now connected newly by joining in the same cytoplasm. But yeah, I think that falls outside of the sort of strict definition, at least, of admixture, kind of in real time.
So there's, if you were infected by a virus today and it integrated into your germline, that would be admixture, right?
Yeah, I think so. That's basically, that would be, I think that would be a nice example of genetic pollution. Whether that's interbreeding, again, might fall a little bit short of the strictest definition.
Oh, so it's by interbreeding. That's part of the definition. Okay. So that makes it hard. So I'm not aware of any admixture in our genome, are you?
Not off the top of my head, at least in that strict sense. But that's a really common occurrence for single-cell organisms, for microorganisms, which is sort of our topic today. So maybe should we step back and introduce? So the paper we decided to tackle for TWiEVO 48 is
47.
Oh, sorry, I'm getting ahead of myself. Thanks.
Next month. Sorry,
thanks, Vincent. That's right. Yep, our four-year anniversary coming up fast. Evolutionary biologists should be always looking into the past, not into the future somehow, but they both come together. Anyway, the paper is a polyploid admixed origin of beer yeasts derived from European and Asian wine populations. And so this was a paper that showed up in PLOS Biology. When was that? Maybe last year? Bringing it up now. Oh, no, earlier this year. So March 5th, 2019. We'll get into some of the details in a moment. I wanted to just highlight some of the authors and some fun personal connections. So Justin Fay, sort of the senior corresponding author, first author of the paper. He and I actually overlapped in grad school. He was a year or two ahead of me in our grad programs at the University of Chicago. And I wasn't really studying, evolutionary genetics until sort of late in the game of my graduate training. And so I was kind of looked up to his work. He was in Chungyi Wu's lab, famous molecular evolutionist, I think potentially now retired or getting close. And in fact, this is one of the cool things out of several cool things out of his thesis work, but he actually had a population genetic test named after him, Fay and Wu's H test that he developed when he was in grad school. So it's kind of one of the fun things I would say about population genetics or molecular kind of quantitative molecular evolution is all of these sort of metrics that are named after the scientists who put together kind of invented them or put together the equations to try to capture. In this case, it's to try to distinguish neutral from positive selection in a way that sort of extends from a different famous population genetic test called Tajima's D. So it's almost like a whole language of genetic testing in population genetics. So if we go down the author list, another familiar name to us at TWiEVO and maybe some TWiEVO listeners, Maitreya Dunham.
Yep.
And she was our guest on TWiEVO 22, E pluribus cerevisiae, from somehow in broken, sort of half-baked Latin, from many yeasts, one. And we talked a little bit about experimental evolution in yeasts and how the single cells could quickly go to sort of aggregates or flocculants, collections of yeast that could drop to the bottom of the tube, and how you might select on that, and what would be the genetic basis sort of in real time. So Maitreya, very established and increasingly influential voice in evolutionary genetics, experimental evolution, and in particular, I would say yeast evolution, given all of her great work setting up those turbidostats, or those, blanking on the name of it now, these sort of automated systems to dilute out nutrients, add in nutrients, dilute out the yeast, and do just continuous cultures of yeast over time as they adapt to things like restricted nutrients and or other challenges that you might throw at them in the laboratory.
It's not a chemostat, is it?
That's what it is, chemostat. Thank you. Yep.
I noticed that Justin is at the University of Rochester.
Yeah, not too far from your neighborhood over there.
But that's where Harmit went to school, right?
Correct. Exactly. Great history of consequential evolutionary genetics coming out of the University of Rochester. In fact, Justin, so he opened his lab first at Washington University in St. Louis and ran it there for many years. And that's just in the last couple of years that he moved his group to Rochester, upstate New York.
Wow. Change of weather there.
Change of weather.
Oof. Because it gets really cold and a lot of snow up in Rochester.
Yeah, St. Louis has at least slightly cool winters, but I agree. He's adding some cold tolerant selective pressure to his menu. Sure.
A colleague of mine retired, I don't know, 5 years ago, and I went to steal some books from his office he didn't want, right? One of them was a little book called How to Make a Phylogenetic Tree. It was written by a guy at the University of Rochester. In the acknowledgments, he thanked Harmit Malik for help. He must have been a student at the time.
It's true. Yeah, I have that book, and I just lent it out to one of the students in my lab. And so, do you have it there? I'm blanking on the author's name.
No, I don't have it within reach.
Yep. But anyway, so that was when Harmit was making this sort of massive transition from his training as a chemical engineer to then into genetics. And so he likes to tell the story of how the first genetics class he took was actually, his role in it was as a TA. And then similarly, he was contributing to books on phylogenetics at the time he was just kind of learning it for the first time, sort of like just-in-time education.
Wow.
Yeah.
Chemical engineer, huh?
Yeah, that's right. Big transition. But Rochester really does have not just sort of a historical reputation, but a growing new one. So with folks like Justin moving his lab, Nancy Chen just opened her lab. So she's another really good population geneticist coming out of Graham Coop's lab at UC Davis. She's working on things like scrub jay populations, where you actually, they're becoming kind of so rare that you can actually follow an entire population over years of field testing. And then she's doing pioneering work to get an understanding of the population dynamics from sort of a quantitative genetic perspective. And so all kinds of cool stuff happening at Rochester in the evolution realm. Good. Yep. Okay. So anyway, getting to the paper. So now we are going to take a little bit of a dive into the origins of beer yeasts. And
so who cares?
Well, it's a fair question. I'm feeling a little bit thirsty over here myself, I have to say, but
I mean, it works. Just keep making it, right?
Yeah. Well, we're talking about a pretty massive industry here. And it's not just, so yeasts are not just deployed, as I'm sure everyone knows, for beer brewing, but also for other fermentation processes. So of course, things like wine, champagne, sake, rice wine. We're now, this is now into pretty major agricultural and recreational sort of industries here. Also extends to maybe some lesser known contributions for yeast, things like palm wines, coffee, cheese, and of course, bread, baker's yeast. So there has been a pretty complicated, as we'll see, and entangled history of the evolution or origins of a lot of these useful yeast species. And I guess, one application or one idea might be if we understand not only where they came from, but sort of what are the key evolutionary changes or genetic differences between them, that you can start to use that understanding or use that knowledge to continue to select in ways that might be profitable. Mm-hmm. And I think that's, not only sort of the fancy latest new style of beer, but, technical things behind the scenes, how fast the brewing process goes, or the kind of characteristics, at what scale you can do it, or under what environmental conditions. The more flexibility you get there, the more possibilities that could sort of open up. I think it's also interesting, I guess maybe from a more academic or historic perspective, what's going on. So, of course, in our last episode, we considered the origins and evolution of a much less appetizing topic, the canine transmissible venereal tumor. And so I think it's kind of fun to think about something that's, not only sort of potentially medical history, but also something that kind of influences our lives as humans, as we've domesticated yeast species for all kinds of food and beverage related outcomes.
So the yeast ferment and produce ethanol, right? In the presence of oxygen. So if you knew all the enzymes involved in that, is that all you think is important to worry about, or would other things contribute? Because you could say, all right, let's take all these enzymes and start mucking with them in some way.
Yeah.
Right.
I think that gets you part of the way there, but I think there's obviously way more to genome evolution or sort of physiology in the properties that go into that. And so, so I think all of the above, and I think there's increasing investments made by massive brewing companies and all the way down to microbrews. Actually, there's one of my favorite stories is from a few years ago, a really small microbrewery that wanted to do one of these sort of hyper local beer batches where basically everything was grown or obtained on site at their brewery. So, they grew their barley, they grew the wheat, they grew the hops, they got the water from there. I'm probably getting some of the ingredients of beer wrong, but and then the last thing they needed was a local yeast strain. And so they actually hired a microbiologist, and I think this might be the Rogue Brewing Company, if I'm remembering right, in Oregon. So they hired a microbiologist who went around the brewery with all kinds of yeast plates, and was, out in the soil in the yard and around trees where a lot of the wild strains originate, and which is sort of an important point for this paper that we're considering. But nothing really grew except on one plate. And that was, they did a swab of one of the brewer's beards. And of course, being a bearded fellow, this guy probably had all kinds of yeast hanging out just from working around the vats and during the fermenting and beer brewing process. And so, they actually decided to market it that way. Like they weren't hiding that they they cultured it off of one of the brewer's beards. I think they called it New Crustacean was the name of the beer, and had it really grosses people out actually. Even though, obviously there's yeasts floating in and out. It's actually a big part of some brewing processes in like Belgium beers where you're collecting wild strains or, kind of taking advantage of these natural contaminations or exposures to add character or flavor in the beer brewing tradition.
Yeah. I once heard a talk, must have been at an ASM meeting, where a guy was a microbiologist. He was interested in the origins of beers. Mm-hmm. And most of it I've forgotten, but I remember he said you can find in Egyptian ruins some old beer in old vessels that are been sitting around, and people have done that and analyzed it and tried to replicate it today.
Yeah, no, that sounds familiar. And I guess maybe that would be a slightly analogous if you could, to the sort of the ancient DNA, right? So that you're not just, and as we'll see today, as we're inferring, again, insights about the history from sampling modern species and trying to reconstruct that through phylogenetics and some related population genetics, having those ancient, if they're really well preserved and sort of cut off from future contamination, you could actually get a peek back into some of what the strains looked like in ancient times. And so,
so he mentioned one that's made by Dogfish Head, which is a contemporary brewing company, and I bought it, and I drank it just the other day. It was horrible. It was way too sweet. Ancient ales.
Interesting.
Go to the Dogfish Head. Of course, if you want to go to their website, you have to put in your birthday because you have to be overage. Isn't that ridiculous? Just to look at a website, I have to put my birthday in.
21 and over. I don't know how they enforce that, to be honest.
No, because you just put any date in, right?
Of course. Yep. Exactly.
Anyway, it wasn't very good. It was too sweet. But
yeah, well, and actually, that might get back to your earlier point that if you just monkey around with the enzymes, would that be enough? And I think there probably there's something to that, but it's really kind of in that process of artificial selection or domestication, where you're kind of using the human palate in this case as your selective tool, and you're saying you're trying out a batch and you're saving a little sample of the yeast so it's not lost or it remains active or viable through the, if it's unfiltered, I guess, through the, depending on how you're actually doing the brewing process.
Yeah.
But then if there's something that you just really love, that it's not sweet or it worked, then that's what you hang on to, right? That's the process of artificial selection. And so that's a concept we've covered a little bit. There's a similar idea back on TWiEVO 7. We were talking about pigeon breeding and how, in this case, the selection is pigeon fanciers who, want feathery boots on their pigeons. And they get that, right? So as Mike Shapiro, one of my colleagues here at Utah, he told us about his fascinating work trying to reconstruct what is the genetic basis, basically, of that artificial selection. And so this has an echo of that now kind of at the microbial level and, not selecting anything you can see because these are microscopic, especially stretching back, hundreds of years in the kind of back to the 15th century, potentially for some of the origins of beer brewing. But you're now selecting on something you can't see, but you can taste. Or there's that sort of experience that comes with it, including ethanol content. And so that then underlies the history of sort of letting evolution, in a sense, do the natural experiments for you. And then you, as kind of the agent of artificial selection, then take advantage of that. What's really, I think, exciting is kind of bringing the two methods together, sort of from the kind of genetic engineering or even kind of bioengineering of enzymatic functions, but then seeing how you can play that off of sort of the history of artificial selection. And so I think having some sense of the evolution is kind of how did it play out naturally kind of gives you maybe a set of rules or a sense of what might be the pressure points, because there's so many, the challenge of bioengineering is there's just so many things to change and to sample. And so how do you prioritize? Because you just can't cover the mutational landscape or to try out everything. And so this at least I think helps to put some definition into how it worked naturally up till now to see how things might go forward.
They have a word here, Nels. All right, so we covered admixture. There's another word in the paper, allopolyploid. So polyploid means you have, every cell has a different number of chromosomes. Is that right? That's heteroploid, I guess. Right?
Yeah, no. So polyploid just means, so polyploid means more than diploid generally, right? So diploid would be you have one copy of your chromosomes from your mother, one from your father. And then if you undergo a whole genome duplication, you become polyploid. And so this could be triploid, tetraploid, even some, especially in plants, things can go really crazy. Here, I think the yeast and Maitreya was talking to us some about this in the polyploid solutions they see from their, even in short time from experimental evolution. Here, polyploidy, or I guess she's mostly looking at aneuploid scenarios where part of the genome is duplicated. Here in polyploid, we'll be inferring a history of whole genome duplications from diploid to triploid to tetraploid in some of the strains that they're considering. Yeah.
And what does allo mean? Allopolyploid.
Yeah. So in that case, I think it's just doing a quick web search here. So the chromosomes are composed of more than 2 genomes, each of which has been derived more or less complete, but possibly modified from one or species. From other species. Yeah. And so that returns to the notion of admixture, right? So these are, I think what kind of distinguishes this, like, for example, from the transmissible tumors that we were talking about on TWiEVO 46 last episode, where they were tracing all kinds of SNPs, single nucleotide changes. Yeah. Here, I think the major events in yeast evolution have been sort of these, like, as people have tried to breed or intermix some of the, and maybe even just to begin with, just thinking, oh, here's a strain that we use to make this wine. Here's a strain from bread or from sake. Let's just mix them together and see what comes out on the other side of some fermentation process. And then in the meantime, the yeast kind of started exchanging genetic material, and you end up with these massive admixtures or like major chromosome sort of donations or rearrangements followed by recombination. And so here we're, like, in contrast to the last story on the tumor evolution, and where it's all mostly, at least what they were measuring, were the point mutations. In this case, it's actually the massive genome rearrangements that might really be driving some of the big events when it comes to then selecting on traits that might be useful in brewing or other fermentation-related scenarios. And so just to put a little bit of some detail on that, so I think the major sort of traits that are beneficial or would be try to keep them around would be fermentation in the presence of oxygen. So this is kind of traces back to the idea of how yeast can switch their entire metabolism when they lack oxygen to fermentation. But if they can continue that in the presence of oxygen, then a nice side effect is that you can get higher levels of ethanol that in other strains would be toxic. And so basically you go from a 3.2 beer to something a little more higher octane, as the case may be. But another, actually, I think really interesting from an evolutionary standpoint, major trait that's important is to be able to compete with other microorganisms in high sugar, low nutrient environments. And this is kind of key, because otherwise, if you're, let's say you own a vineyard back in the 1300s or something like that, and you go out and you harvest grapes that you've been meticulously growing and keeping safe from birds and other predators, then all of a sudden, you go through all this trouble, everyone's mashing up the grapes, dancing on the grapes, all of those great traditions. And then just as you're hoping for the yeast to start to ferment and make this into wine, if some mold or must, grape must, I think is a fungus that could just come over and just make the whole just take over, taking advantage of the high sugar from the all those grapes you just smashed, but then not ferment in a way that makes anything palatable, but just it would go rotten really quickly. And so that, I think, is another key advance that actually might really trace to the origins of wine, which I think has been inferred to 9,000 years ago. And maybe also just to say that this, the work both on yeast and just, in general, so we're talking about Saccharomyces cerevisiae, which is familiar to scientists because it was co-opted as the genetic model system, the sort of scientist yeast, but is also seen as brewing conditions, brewer's yeast, and also, or brewing conditions, and also baker's yeast, which is sort of also a well-known trait. So all kinds of, even within the species, all kinds of useful traits for fermenting or using in both brewing and beverage making.
How do you think that first batch of wine got made 9,000 years ago? You think it was just an accident and someone drank it and said, "Hey, it's pretty good"?
Yeah, I don't know it's a great question. I don't know the anthropology there, but that seems the most likely to me. That was an accident, right? Yeah. And that there was just some yeast hanging out nearby, and instead of it going bad, or I guess it did go bad, but that bad can be good in this case. And then that got people onto then not only sort of depending on those random events for the grapes and yeast to come together, but to actually do it on purpose, and sort of the human intervention coming in, human associated interventions that then quickly picked up.
Yeah.
And continue today.
So this aloe polyploid was used, they say, besides S. cerevisiae, another widely used species is S. pastorianus, which is an aloe polyploid hybrid of S. cerevisiae and S. eubayanus. It's used to make lager. Emerged in the 15th century in Europe. It emerged from a strain closely related to strains in North America and Tibet. Oh my gosh, it's just so complicated, isn't it?
Yeah, no, exactly. And I think that might be the take-home message. At least that was mine from going through some of the phylogenetic reconstructions and the population structure analysis. But yeah, so I think those cases around the 1500s when beer was really starting to come into fashion.
Yeah.
And some of the traits there were being able to ferment at low temperatures. And I think, this was obviously kind of like wine cellar or like catacomb brewing or something like that below ground. But as that was taking off, the ability for these yeast to conduct fermentation, but at lower temps, that was what they were really looking for. And that's where somehow these hybridization events, I think, again, my understanding, mostly happy accidents of things coming together, and then when something worked, you'd hold on to it as part of that artificial selection.
Yeah, so the history is muddled, right? And that's one of the take-homes of this, that we're not quite sure because over the years people have done different things, but they didn't always save the strains, right?
Oh, yeah, exactly. Yep. Yeah, and how much possibility there is to kind of match this with historical collections, I don't know. And to say there's all kinds of other studies happening right now, not just coming out of Justin Fay's lab, but several groups that are really interested in this from both genetic and evolutionary perspectives. So all kinds of emerging work, and kind of stay tuned, I would say, to see how much unraveling or kind of correlations can be made. But this is definitely a cool first stab or continuous stab at it, using a lot of new genome sequencing, actually, as we'll see.
I like the way they say ale and lager strains and ale and lager alleles.
Yeah, that's right. It kind of reconnects it to and it is true that once now, and especially now, as things have become increasingly specialized, brewers are really getting more and more particular about, based on the type of beer you're brewing, which kind of yeast you'll use. You don't just, and then kind of combining that with sort of increasingly sophisticated choices about the strains of hops and how you get your flavor profiles, the bitterness, et cetera, et cetera. It's all kind of coming together in new and more complicated ways all the time.
Have you ever made your own beer?
I actually haven't. I've grown hops. I have some hops growing right now in a garden. And I also have been keeping bees. So I've got honey. So I've got like two potentially good ingredients. But I have, how about you, Vincent? Have you done any?
I have not. No. But what are you growing hops for? Were you thinking of making some beer?
I didn't really think that far ahead. I just thought it'd be fun. They're kind of viney. I had a fence that I wanted to cover. Yeah. And I actually just like picking up some hops and just for the kind of the resiny, just kind of bitter smell. I think they're just kind of cool. So, but as those hops have become established in the last few years, maybe there is some beer brewing in my future. We'll see if I ever can carve out the time. Could even have a podcast on the topic. Yeah, for sure. There must be hundreds of them, actually, now that I think about it. But
yeah, there are plenty. Yeah.
Oh, yeah.
So what'd they do with your Nels?
Yes. So anyway, kind of as we've already been kind of dancing around a really complicated history. And in fact, they're already narrowing in. So they're all of, as you're saying, like they're deciding to focus on some of the ale strains. And these themselves already are known to have complicated histories of admixture, involving, cerevisiae species that they're all within strains within the same species, cerevisiae, that have been used for grape wine, champagne, sake, et cetera, kind of down the list. And so what they did here to advance sort of the evolutionary view of where some of these modern beer strains of yeast have come from is they use a combination. So there's already 430 at the time of this work, 430 publicly available genomes of yeast. And this is, again, mostly cerevisiae. So it's already focusing down on that one species. And then they do, or they sequence 107 new ones. And the idea of putting this together, expanding it, is that they are making some choices about strains that are known to be either wild, so they would be sort of assumed to be ancestral, stretching back, versus the ones that are domesticated, and then to have sort of some comparison points with ones that are found in the clinic. So these are people, oftentimes immunocompromised people showing up with yeast infections. And then also lab strains, sort of this very recent last kind of 60, 70 years history of bringing some of the yeast strains into the laboratory. And so with those new ones, they can, along with the sort of collection now of more than 500 yeast genomes, start to try to infer some of these points about when did the hybridization events or the admixture events, what were the main ones, the kind of transitions to polyploidy. Can you sort of trace or get a understanding of how these strains have emerged over the last several hundred years as humans have really taken them up as a valuable commodity? And so of those 100 or so strains, new ones, that they picked 50 specifically that are brewing and baking strains, and then another 65 that they say are from diverse sources. And so these are, for example, the wild collections, the clinical lab, et cetera. And then if we look at so then you've got all the genomes, you can start to do, to build phylogenetic trees. And they're not, so we've talked quite a bit about phylogenetics on TWiEVO. And what's interesting about this is it's sort of is in a different way of assembling a phylogenetic tree. So the branch lengths are based on genetic drift. And that is, so they're inferring, again, using sort of some population genetics tools, as populations are bottlenecking, sort of the collection of alleles that or genetic variation that just makes it through. And so it's not like you're selecting on the perfect mutation that allowed this enzyme to do better, or the loss or judging conservation because you've lost this deleterious allele that would crash out the population because it's not working well. It's sort of the marbles out of the bag notion of things that just made it through population bottlenecks. And part of the reason or motivation for doing that, or to kind of take that slightly different view, is because they already really knew that admixture was going to be so sort of rampant in the data sets. And so what that means is if you just picked one or several genes or a cluster of genes and then tried to reassemble a tree based on that, but the fact that there have been so many sort of horizontal donations, hybridizations, admixture kind of events, it's really hard to infer sort of vertical inheritance through that technique. Yep. And so, you might think, oh, great, I have this gene from this strain, but in fact, if it was donated yesterday from another strain, those kind of relationships on the tree would be all scrambled. And so they're trying to, take kind of a different tact to get around some of those issues. And then from there, what's different about this tree than many that we've looked at, or many that you'll see in evolutionary papers, is that they draw on these red lines that sort of break the rules or cross between the tips of the branches. And here, this is from their analysis of actually trying to chart when and where did the major admixture events happen that lead up to the modern ale strains or the modern lager strains of Saccharomyces cerevisiae. And so if you look at figure 1, I would say some of the take-homes from that are that there were kind of 2 key donations from other strains, sort of independently, or admixture, sort of these major genetic transitions. And so one is from the Asian strains that were originally derived in the sake making process that then show up kind of at the root of the ancestors of the lager and the ale strains, which are most related. And then the second, although I have to be honest, I'm not totally clear on this, that go from the European or wine strains, which are more closely related to the lager and ale strains, maybe not too surprisingly. Those actually show up in some of the modern clinical and lab strains.
Yes, weird. Yeah.
Although, I mean, it is weird, although I guess, that sort of intimate association between wine drinking and then things that in immunocompromised scenarios among patients, right?
I guess you could get a yeast infection from your wine, right?
Yeah, right. Maybe there's an echo of that. Obviously, so, and just to say, in terms of the notion of clinical strains, so cerevisiae clinical infections are pretty rare relative to some other more well-described pathogenic strains or strains that can become pathogenic when they're exposed to humans. So the Candida genus, as opposed to the Saccharomyces genus, is probably the major current contributor to clinical issues in our species. But still, that does show up. And so, yeah, maybe that kind of close association maybe has something to say about that. Okay, so I think, what you see then is some, and, maybe echoing again on some of the other sort of geographic phylogenetic trees that we've looked at, including last episode of TWiEVO, you see, the Japanese strains are most close together. The European and ale strains are together, whereas the Asian sake strains, those are the ones that are sort of breaking the rules, I would say, in terms of or at least in donating or crossing the genetic boundaries in admixture scenarios.
So the Japan strains are for sake, I guess? Or is it wine or
yes.
beer? Do you know?
Good question. Yeah, so Japan 1 and 2 versus the Asia sake.
Yeah. Yeah, I don't know what they are.
Yeah, that's a good question. Yeah.
I guess they're not labeled. Well, we have ales, we have just beer, which is, then there's, I guess the Mediterranean / oak is from a tree.
It is. And so that outgroup, that branch is representing the wild strains. And that's sort of the not quite, but in a sense, outgroup is the wrong word. But those are we'd infer that those strains they're now obviously the modern descendants, but that those were the ones that were sort of the original ones that you started with, as humans were starting to take these in, and that's where you would find them. And in fact, I think not just the oak tree, but also the so I think some of the original ones, and you do see that inferred in that deep branch, the European wine, were associated with grapevines. As well from wild sources. Yeah.
So this tree is rooted, somewhere beyond China. There's a branch that goes to China. Then there's another branch that goes everywhere else. That splits off. One part goes to Africa, Japan, Asia, sake, clinical, and lab. And then the other branch, you have Mediterranean oak, which seems to be ancestral to European wine, lagers, ales, beer, and baking. Right. So two distinct branches from the original ancestor.
No, exactly. And then I think the take home is that with lots of donations between, and in fact, they do some, they use some population genetics tools to try to estimate how much of a contribution would show up, like in a modern, if you just pick a modern beer strain, an ale or a lager strain from that collection that's sort of, that is summarized in the tree. And in fact, I think they should say, for one example, that there's a 40% kind of genetic donation, if you will, from the sake strain into the beer strain, and that the other 60% comes from wine strains. And so we're really talking about like major genomic events here, right? Where it's big recombination across long distances. And they're also, so one thing that you would, depending on when these admixture events happen, you might think, okay, well, if you got a strain that you really loved, that then you would start to do all kinds of back crossing, right? So after the admixture event, and that you would then maybe start to, if there are actually traits, specific traits that you're really narrowing down on as you continue kind of artificial selection, then what you might predict is that you'd actually have really small contributions from different sources as the kind of blocks of recombination broke up, and you were just selecting kind of at a more high resolution rate. And in fact, they don't see that. So they don't see much evidence of backcrossing after the admixture. It's these major recombination events, or these major contributions, are still sort of the rule rather than the exception for a lot of these modern cerevisiae strains used in brewing. And so I don't know if you came back in a million years, depending on how specialized yeast brewing becomes, or how much sort of bioengineering interventions come in, maybe that will be a different story for the if you just look kind of genome wide at the sources of genetic variation within these things.
Yeah, I think also companies are making strains that are proprietary. You'd never be able to get the sequence, right? So,
well, that's right. Yeah. So, there, I wonder if you could do some biohacking. So actually, if, if you can,
get them out of your beer, right?
Right.
Does
No,
beer
exactly,
still have live yeast in it?
It depends on some. Yes.
Most
of them, no.
pasteurized, right?
Correct. And so in the major breweries, especially the ones that want sort of that proprietary thing, but there's probably still yeast DNA in there. And as genome sequencing techniques get more and more democratized, maybe you could do some biohacking and see what they're really using. Honestly, I think my take home from this paper is that my sense would be maybe there'd be a couple of surprises under the hood of those genomes, that mostly it would look, we don't have that resolution, right? It just looks like you have kind of these major contributions, and some of that sort of perceived proprietary or sort of specialness, it probably is a relatively generic yeast strain, at least from what we know about it from the resolution of the genetics today.
Yeah.
Yep. Okay, so then we already hinted at this notion of the polyploidization of these, and so they wanted to address that issue. And are there kind of patterns that emerge from looking at so not only are there these sort of whole or massive kind of chromosome level genome donation events with big recombination, but then the other question is how much whole genome duplication has happened, or polyploidization. So you go from 2 copies of your chromosomes, diploid to triploid to tetraploid. And so figure 2 summarizes the data they got in a few cases. I think they analyzed 105 of their genomes. So I think those might be the ones that they actually did the sequencing on. And basically the tool they used was to ask, what are the allele frequencies? So it's, so you're actually not doing anything like you could imagine doing something like cytogenetics, where you're just physically counting, like doing squashes and physically counting. So they wanted to use some population genetics. So instead, to infer polyploidization, they just look at the frequencies of alleles at heterozygous sites in different strains. And so if you just kind of make the assumption that you have, gene allele A from your, I was going to say mother and father. I think I should say alpha and A are the 2 mating types of of yeast here. So if you look at the A allele, it might be, it might have 1 SNP or nucleotide change, whereas the alpha allele might have a different SNP. And so on average, you'd say those will probably be at 50-50 in heterozygous positions, meaning that you've got 1 from type A, 1 from type B. Mm-hmm. And so they show that pattern, and I think most of the strains actually fall into that category, maybe about half of them. However, they do see some that where the patterns of, as you count up the frequencies at a lot of different allele and kind of you actually start to see more complicated patterns. And so one class are the triploids that they're inferring, and this is what they're seeing is actually that a lot of those alleles are at 1/3 frequency instead of 50-50. And so that's consistent with having 3 versions. And you have allele A, B, or C. And then they also see what you would expect in another class. So, and I think for the triploid case, that's 23 strains. And then they chart out 28 tetraploid strains. And here, it's an even more complicated pattern. So you see alleles at 25, 50, and 75% frequency because there's 4 different sort of possibilities, and then the combination that sort of falls into kind of the old Mendelian bins in a sense. So there could be false negatives because this big assumption you're making is that you have enough kind of heterozygosity to make these sort of genome-wide comparisons. But anytime you see I think their patterns are pretty clear. You can see the almost clear binning on their graphs of allele frequencies there. And so kind of a clever way of, you go through the trouble of sequencing the genomes instead of having to do chromosome squashes or, kind of those techniques to just sort of milk the data to actually make inferences about the entire genome structure.
Mm-hmm. How many alleles did they look at? Do you know?
I think it's in the hundreds. So just kind of squinting at so each red dot on their graph, those are, it's a heterozygous site that's being typed. Mm-hmm. And my memory of yeast genome, Saccharomyces genome, is there's about 5,000 genes total. So that would be sort of, I think, the ceiling on number of alleles, a number of genes, and then you could still have multiple alleles per gene. And so there's not limitless differences, but into the thousands, potentially to type for kind of overall patterns. Yeah. And that nicely emphasizes too how you can so if you took any one point, you can't really make anything out of it, but doing it genome wide, where you can start to see new patterns and then draw inferences from that, sort of kind of the bread and butter of population genetics, I would say. Yeah,
you can download the chart that they use to tell you exactly how many alleles they looked at.
Okay. Yep.
It's on a sharing site. Yep.
Yeah. Yep. So from there, you can kind of start to overlay, of those heterozygous patterns, how many of those come from sort of admixture versus afterwards. And I think, and so there's another level of analysis here to ask about patterns of shared SNPs, depending on if you can infer where these things came from. And so this gets into figure 3, which is a kind of a complicated one. I don't know that we'll go do it too much justice here or go deep into it, but basically, this is, and I think we've maybe looked at one or two papers that have sort of a simpler version of this kind of analysis. So if you just kind of scrunched on the left side of figure 3, are kind of population structure. So it's color coded based on the population structure, the genome contribution per strain, basically. So most of the Asian sake strains are orange color coded, and you see that's infiltrated by a little bit of a dark color there that they can't necessarily trace exactly to those strains. And that would be sort of an example potentially of like a recombination event in those individuals. So like each line, very thin line, is a single strain that's being analyzed. Based on the ones at the bottom that are completely orange, those are sort of all of the genes could be traced to that. So it's sort of a purebred, in a sense, sake strain. And then you can start to see signatures of admixture as you kind of go through the list. And then they're doing sort of this SNP kind of homozygous versus minor and major allele contributions, heterozygous alleles, and to try to get a sense of when, during the evolutionary history of admixture, do you sort of diversify farther, meaning that you go from homozygous states to heterozygous states? And how does adding an entire, a donation, which should bring in a lot of heterozygosity in one go, if it's a distantly related species, how did that influence the evolution? Can you kind of get a sense of how many changes have happened before and after? And I think my sense, I don't know if you read it differently, is that it's a little bit of a wash. It's hard to tell any sort of major kind of take-home messages or patterns, just because there have been so many exchanges. And you have such high levels of heterozygosity that the diversity, I guess what they're trying to say, at least to tentatively conclude, is that a lot of the diversification in the differences between any given single strain probably happened around the time that you were undergoing these major genome duplication events. But right. I think it's hard to put down too firm of a kind of global conclusion there.
I agree. Yeah.
Yep. And then they do note, and I think it's kind of interesting, is that there are some, so in the simplest kind of scenarios, you might say, oh yeah, there is this big admixture event, and so some of the heterozygosity, is from a different allele from one, showing up in the strain genome with another allele from the other, from that hybridization event. And yet they also see all kinds of, again, diversification that doesn't match any model, where you could say, oh yeah, you took this guy from the wine strain, and you took this allele from the wine strain and this allele from the sake strain, and now they're together. There's no combination that could make sense from a simple event. And so here they're actually suggesting that there's one of the kind of intriguing possibilities is that it was just donated from something that's missing from the data set. And so because there's 500 samples here, relatively decent sampling of genetic diversity in Saccharomyces, that there could be extinct populations that donated. These would sort of be like Neanderthals donating to the yeast genomes, but we don't have the bones of those guys, right?
Yep.
That might be a little too loose of an analogy, but I think you get the idea. There could be extinct populations that are now extinct that made genetic contributions, and they sort of live on in these artificially selected strains. Yeah.
There's quite extensive extinct stuff according to this, right?
Exactly. Yeah.
Most of what we are seeing is extinct, as we don't know where it comes from. So it's kind of interesting that a lot of this has been lost, right?
Oh yeah, and I think it highlights that, and this is one of our kind of key ideas that we've been trying to share on TWiEVO, is that evolution is often about all the things you don't see, right? And that we ourselves are sort of not even the tip of the iceberg of the genetic diversity that came before us, for example, in our species. And so it's really hard to, like, the things you can't see, it's really hard to weigh the sort of impact of them, right? And I think,
and we're inferring what we don't see, right? We can't see it, so we have to infer it.
Exactly right. And so you're not as confident of it because of that. It's something, but I think that's really most of the energy and action of evolution is in that space of the unknown or the extinct or the things that all the failed experiments for the few that made it through.
Yeah, that's right.
Yeah. Yep. Okay. So then the last sort of push here was to actually do a little bit of a deeper dive on a few of the strains. In particular, I think this is a couple of the ale strains, if I'm remembering right. A German ale strain and a Belgium ale strain. To use long-read sequencing to start to phase the haplotypes. And that sort of jargon means that you're using, so up until now, all of the genome assembly or the genome analysis is based on mostly short reads. So these are like, in Illumina is probably the main mover on that. I actually didn't have a chance to delve into the methods of how they did their 100 or so new genomes. But basically you're doing short read assemblies.
Yeah, and so Illumina,
yeah. So one of the compromises there is that you're kind of it's like a jigsaw puzzle almost of reassembling the genome with small pieces. And so you can't say that one mutation on one side of a chromosome is actually associated or phased with one later in that chromosome, because there's no kind of chain of custody across the chromosome, because you're just assembling tiny pieces. And so instead, using long-read sequencing, which is advancing really rapidly in exciting ways, you can actually make massive single reads with enough sort of precision that you can reconstruct not only the whole picture, but actually which shared variation across, or a haplotype across. And they use this for a couple of the strains, one of the German ale and Belgium ale, to see whether they can make out sort of some other patterns in terms of the recombination events that could be happening between again, stepping back to the big admixture events, the European wine strains, the Asian sake contributions. Can you actually start to see recombination between them on the same chromosome? So you're now starting to scramble the alleles through recombination. And so, again, I would say maybe not the most, definitive, in conclusion, or sort of smoking gun for massive, signal for, take home concepts or anything like that. But I think what they came home with was relatively short haplotypes, which is implying a lot of recombination between the original strains. I think that makes sense given, how yeast conduct their genetic business. There's a lot of recombination happening, big populations, short generation times. But there is some useful information that it can actually be teased out of really carefully phasing these haplotypes. And that is that based on the recombination measurements, you can make actually time estimates from the time of admixture. So you've got this, 40% contribution of the sake strain, 60% contribution of the wine strain. And then based on how short the haplotypes are, you can start to say, how long has it been since these things just showed up together just as massive blocks? And so they kind of walk right up to the edge of the cliff on that, but then I don't think they actually put down any suggested time estimates. And that, again, we've talked about mutation rates and how do you sort of get to a molecular clock. That can also be a pretty tricky business to really put your thumb down. But I think at least I took home from that part of the analysis is that you have a lot of sort of genetic recombination, a lot of scrambling between the strains after the admixture events and as they continue to be used for brewing.
Mm-hmm.
Yeah.
And they say that there's a more recent admixture of some versus others. Lager and the two ales were older.
Yep.
That's about all they could do. It's
That's
relative.
right.
No, yeah, as you say, no numbers, just relative. Yeah.
Yeah. Yep. Yeah. And so I think this is sort of an opening contribution, right? So now that you can do genome scale analysis into the hundreds, or in this case, 500 comparisons, and then a really nice sort of complementary contrast to our last episode where those tumor genomes were analyzed basically based on SNPs, point mutations. Here we're way at the other end of the spectrum of trying to analyze these these yeast genomes where there are these huge kind of chromosomal contributions through the process of admixture, and as these strains have been intermingled, and then they've also become entangled genetically as well.
I think, though, the other is an interesting question. If you took some of the key enzymes and look at SNPs in those to see how they have changed and evolved, right? That would be curious to look at.
Oh yeah, no, absolutely. And I think that's ultimately one of the goals. And so, but that's tricky too, right? So it's like if you just start there and there's tons of variation, and then you say, okay, well, what is, a lot of people would love to know this, right? What is so if you could connect sort of the phenotypic outcome of artificial selection, which is a yeast strain that's really great, reliable, works every time, can under different conditions, robust, you end up with the product that you want, whether that's beer, sake, wine, etc. If you could take that phenotype and correlate it or associate it really at high resolution with the thousands or millions of genetic changes that you might type just by doing a genome. And so I think maybe what this does is it just starts to provide a bit of a scaffold to say, like, what are the major contributions to start to dissect that apart? Because, making those associations is a bit easier said than done. Another really cool strategy, I think, in addition to this sort of inferring the past, is to actually kind of make a new future. And so, this is happening, I think, in many labs. I've got a science buddy, Chris Hittinger. He's at the University of Wisconsin-Madison, and they're doing some forays into experimental evolution. So it's basically you team up with brewers as yeast geneticists, and then you actually have like professional tasters who are taking batches of beer, and you've maybe made a few genetic changes, or you've tried to really restrict a lot of the mutational differences in that process, or your highly related strains, and then getting them to go on record as to phenotype, this is really good. And then to actually be able to trace that as another way of kind of coming at the question from the opposite angle, which would be, or the other direction, which would be to look at really small changes and try to see how they might be influencing people's reactions. Yeah. So lots of potentially cool stuff. And I think this one kind of comes at it from that really classic kind of population genetics, and to really start to get a handle. And I think, as we've been saying maybe repeatedly now, it's complicated.
So I like their thoughts on where these strains are originated. They say a crucial piece of information where European wine strains were domesticated. So they have two theories. One is a European origin, and that's because they have these Mediterranean oak strains that are related to European wine strains. So, okay, that makes sense. But it's also possible that, and I like this, the Mediterranean oak population is a feral wine population, wild wine yeast. And both European wine and Mediterranean oak are non-native, but they came from Asia. And so they have some evidence that there's an East Asian origin of all the domesticated cerevisiae strains, including European wine strains.
Yep.
It'd be quite interesting, right?
I totally agree. And it's kind of adds another layer of complexity since the plants that humans were moving around the world during, the last 500 years or so, also like that starts to become part of the story as well. Yeah.
So they say European strains could have originated in East Asia. They were admixed with local strains there, sake, rice strains, and or European strains were domesticated in Europe from this Mediterranean oak population, and admixtures came with strains that came from Asia by way of the Silk Route. So you can't tell because of the extensive admixture where these things originated.
Yeah, and this actually kind of contrasts the last kind of evolutionary inference or reconstruction from that canine tumor situation, because there you have, I think a difference is that you have these dog strains from around the world that are actually pretty geographically isolated in some sense. There's still obviously all kinds of pet trade going on, but you could make maybe stronger inferences, right? So they can actually, in that case, they were inferring like dogs moving over from Europe to North America to Africa, back to North America to Africa. I mean, it had that level of resolution. Here, I think it's a bit more, it's harder to do that based on exactly.
So if you want a take-home message, the beer you drink today, the yeasts used to ferment that are descendants of strains used to make European grape wine and Asian rice wine.
That's right.
I wouldn't have guessed that.
Right? Yeah. No, I agree. Yeah. It's kind of a grab bag of two very different fermented products. Yeah, no, I agree. So related to, but not identical to a mix of those two. Yep. Yeah.
So would you say, Nels, most of the work here was computational?
Oh, yeah, definitely. So, I mean, we've kind of talked about this a little bit before, but that notion of just saying, we'll sequence the genome. Yeah. We're so far down the road in that technology. That's, that sentence, it's delivering on that is a lot easier said than done. And so that's still a pretty big contribution is to make careful choices about which 100 or so strains you're going to sequence. That can now, technologically, that can be, the sequencing itself can be crushed in a day or so. But then the analysis is really, the annotation, the analysis, that is a bit of a heavy lift. Yeah. And then from there, we're talking about pretty sophisticated population genetic treatment, trying to squeeze out some of the evolutionary past or population structure using quantitative genetic tools to do so. And I think there's a lot. This is a really nice demonstration of how far you can push it. I think we've also seen really cool studies that sort of borrow from different techniques or fields or expertise. So again, so cytogenetics, right? To confirm physically that you are tetraploid would be another way to do it. And so, but it kind of shows how far you can push on just the sequence analysis alone as well.
And I just want to remind everybody, including you, Nels, as a postdoc, I did the sequence of poliovirus RNA, 7,441 nucleotides. Took me 1 year.
Yeah, I love that story. So you were doing Sanger sequence. Was this like how are you doing that sequencing? Was it like shark-tooth comb and bands? And
extracted RNA from particles, made DNA with using reverse transcriptase. And then I cloned it into a plasmid to make more of it. And then I used Maxim Gilbert sequencing.
Before Sanger. Yep.
Fragment. So I would cut a piece of the plasmid out, say 800,000 bases, right? Purify it, label the ends with radioactive phosphorus, and then cut the DNA with a restriction enzyme to liberate the two ends, because you only want to have one end labeled. And then you subject it to chemical degradation that's specific for each of the bases. And then you get a population. You do one hit per molecule. Yeah. And then you fractionate them on a gel, and you get, smallest ones at the bottom, and then each one is a base longer, and you can, by comparing the 4 lanes, 4 chemical modifications, you could say what base it was. And I should just read up the gel. I used to love doing that.
Yeah, that's cool.
So I would read it and write it down on a piece of paper.
Yeah.
Then I would type it. We didn't have personal computers. We had these terminals linked to the MIT computer.
Yeah.
That you would you have a telephone you would stick on an acoustic coupler and dial in via a modem.
I love it.
And then I would upload the sequences, and then we had programs that would overlap them, make contigs out of them.
Mm-hmm.
And it just took a long time.
Oh yeah, that's some heavy lifting. What would be the most sequence you could get like in a day, or in one go, I guess, of the reaction?
So in a day, you could do reactions and run it out on a gel, and then expose it overnight to film. So
Yep.
if I used to run these big gels that could put 15 samples on them. So I could sometimes on the best gels I could get 400 bases red.
Oh wow, yeah, yep.
So I could get a reasonable amount.
It's a good day.
But I only did 2x coverage,
No, I hear you.
I got both strands, because that was all you could do. And I had nothing to compare it to.
That's right.
There's not
Yeah,
much
there's
else. There was no database to be said.
Yeah, you don't do the blast search, right?
It was one of the first viral genomes to be sequenced, and so it took a while for others to come around. That's why we worked on one virus, because you couldn't do hundreds and hundreds like you can do now, right?
Yeah, that's right.
It's a totally different way of doing science. You can compare thousands of polioviruses, whereas I had one, and I said, okay, here's the protein, and let's play with it,
Yeah, no, I hear you. But, we're doing we just did a coronavirus discovery in zebrafish, and obviously much more efficient. But what's crazy, and it gets to that point about the database searches, right? So it took us a little while to actually get the full sequence. We did some 5' race, some 3' race to get the whole sequence. But then once we had that, and it's about 8 kb or so, kind of a standard 8.5 maybe standard for coronavirus, very distantly related to poliovirus. And but you're just like you're saying. So of course, the first thing we did was go and do a blast search,
right.
And slightly more involved. So we started trolling the sequence read archives of other zebrafish data sets, and we were actually so the postdoc here who's doing the work in one afternoon in one search was able to pull out in someone just deposited this for a different study, not looking at viruses, but hiding in that data set was actually the full assembly of that virus genome in some places cool. To 165x coverage.
Wow.
Just from the raw sequence. Yeah. And so that's an example where like a blast search in that, sort of existing sequence that might not have been analyzed in that way, but it's it's like at your fingertips potentially. Yeah.
Did you enrich for particles?
No. So that's the thing that's really amazing about it, right? Is that it was not enriched at all because the study was looking at the zebrafish RNA, not any microbial stuff. And so this is just sitting in the raw reads. And we think what that implies is that that's a really immunocompromised or susceptible strain. And so it could be a cool kind of 2-for-1 discovery where you can assemble the entire new virus genome, but then you have an idea that actually this is the strain that might be the most sensitive to it. And so we're kind of following up on that idea now.
So your work was just transcriptome, right?
Correct.
So you just extracted RNA and sequenced it, right?
That's right.
And then did you computationally deduct the host sequence?
Exactly right.
Because you don't want to blast the whole thing, right? Yeah.
That's right. Yeah, to save some time. That's right. Yep.
Neat.
Yeah.
All right.
Lots of possibilities. All right.
We have 2 short emails. Let's do them.
Hit the mailbag here. So Trudy writes, might be an interesting paper for discussion on TWiEVO, and she shares a link. This is a great one. So Trudy, we're on the same wavelength here. This is a news piece that relates to actually a couple of papers that came out recently. And so the title of the article, news article that was just from a few weeks ago, Scientists Glimpse Oddball Microbe That Could Help Explain the Rise of Complex Life. And this is a sample called Loki archaea, and kind of a splash a few years ago from some labs on the DNA analysis. So these are the really deep sea vents, extremophile scenarios for life. And some of that sampling and then just the genome sequencing, there are some really provocative ideas about potentially stretching back to the origin of eukaryotic cells and what are the contributions of archaea versus bacteria and so forth. super controversial in part because no one ever had these organisms. That's now changed, or it's been proposed that changed, and these things have been cultured now by several groups, I think mostly Japanese groups. We were going to do one of those papers, in fact, for TWiEVO 47, but then Vincent pointed out that actually, stay tuned. They're onto it on This Week in Microbiology, TWiM.
Yeah, this is, as you say, a bioarchive manuscript, but Nature is writing about it because it's so interesting. And so one of our listeners sent it to us a couple of weeks ago, so we did it on TWiM, and that'll be out on September 19th, which is 2 days from today. And this TWiEVO Mayor will likely be out shortly before or after that. So,
yeah.
Trudy, you can check it out on TWiM. It's a very interesting piece of work. It took them 12 years to culture this.
Yeah, isn't that wild?
Lochiarchaea. Yeah, It's very cool.
Yeah, being able to grow an extremophile. This is not like baker's yeast or budding yeast, right?
No.
This is some sort of high difficulty microbiology here. No, I look forward to listening to that. That's haven't taken a deep dive on it, but I'm glad to hear you already did. On TWiM.
Yeah, and Michelle Swanson got in touch with the authors and got some commentary from them as well via email. So that was fun.
Oh, cool. Good idea.
The cool thing about that paper, I told someone, they're not like just working 12 years on a project and not doing anything else, right? Because you can't
drinking coffee while you wait for it to divide. Yeah.
But, they're at a place in Japan where they can take risks. They are at the Institute for Extra Cutting Edge Science and Technology Avant-Garde Research. That's the name of the institute.
That's pretty wild.
Isn't that cool?
Yeah.
So they say, go do something cool.
Yep.
And we have an email from Anthony who sends a link to a PLOS pathogens paper. He writes, it seems that red hair isn't the only thing that Neanderthals gave us. Perhaps this might be interesting on TWiEVO. Dr. Elde was the editor.
Yeah.
Tell us about this, Nels.
No, this is great. Yeah. Thanks, Anthony, for sending this along. So I don't know if we'll get to it. So the idea that it's really cool, and thanks for making that connection. A parallel idea, so we've been talking some on TWiEVO about how Neanderthals, through introgression, ancient introgressions, have donated potentially some alleles that could have favorable antiviral properties for some of the modern viruses we face today. At the same time, of course, if we think about how viruses travel, in addition to or with hosts, or kind of locked in these genetic conflicts, perhaps there are also donations, in essence, of ancient viruses through that lineage. So back at the time of introgression, could there also be virological donations, in a sense, and then sort of a parallel evolution coming through a virus lineage, in this case the papillomaviruses, tracing through.
Mm-hmm.
So yeah, no, very interesting. I don't know if we'll get to it, but I did want to mention for a full episode, but I did want to mention so I've been serving as an associate editor for a few years at PLOS Pathogens. And this one actually had kind of an interesting, slightly controversial turn after it was published. So there was, and actually, if you go to that link that we'll put up on the website, there's a couple of reader comments. And one of them, it's really short, kind of a glib comment, which was, oh, this work was already published. And so, that's an important issue, actually, to consider. So this article has already been released, meaning that it was published by a different author earlier. And so PLOS Pathogens takes those comments pretty seriously and instigated, I wouldn't say like a formal investigation, but definitely like a follow-up, where contacting both the author, or contacting the authors, me as the editor, and the reviewers to weigh in on that comment. And in fact, in this case, it's sort of a relief. So we had already considered that exact sort of possibility that came up in the review process, where there was some overlapping work that had been previously published. The authors actually weren't trying to hide that, so they included, not only did they cite that work, but they also actually included it in the introduction and discussion and tried to sort of frame their work next to what had already been proposed. And actually in the review process, a couple of the reviewers sort of flagged that as a potential issue. And so in the sort of the resubmission process or the revision process, we did ask the authors to be even more explicit about that. And so luckily we were had already crossed that bridge or covered that issue and returned the revision to the reviewers to then weigh in if they thought that it was enough of an advance given that sort of overlapping work. And in fact, they all agreed it was. And so it was ultimately accepted for publication. But so I guess the good news is that PLOS and other journals and editors and reviewers take that notion very seriously of, making sure credit where credit's due and not kind of copying or overwriting previous work, I would say. So that's the good news. The bad news is there's so much, increasing scientific progress on all fronts and overlapping fronts that sometimes authors don't have the best intentions and can get away with some maneuvers like that. And so the system doesn't always work. I think in this case it did. And then you can read the paper more carefully and actually weigh it. The authors weigh it there, and you can read for yourself the other publications and see if you agree that was sort of properly handled.
It's interesting.
Kind of a fun side story there.
This article has already been released. That's pretty funny.
Yeah. Well, and it's kind of, I think fair enough, but you also I wonder how long it took to read that comment or to write that comment relative that it took us to kind of go back and touch all the bases to make sure that we had addressed that in a good way.
It's funny. It's odd the authors didn't bring it up, right?
That's right.
Very good. How about some picks?
Yeah, let's move into our science picks. So I have one. It's I hope this works as a Twitter link. So I've kind of been getting excited about our last two episodes where we're using phylogenetics to kind of infer thousands of years of activity with sort of human intervention. This is sort of maybe a fun wrinkle on that. So this is about 500 years just tracking population fluctuations in the 10 biggest cities. And so it's on Twitter. I was trying to look, I kind of ran out of time. I was trying to look for the original source. I think it's at least partially sourced. This video, I guess, or clip of the graphic, sort of dynamic graphic. What it does is it picks the 10 biggest cities from the 1500s, and then it counts through the years. It shows a little map so it's first of all ranking the top 10 cities, and so that gets updated in sort of real time. So you can kind of see if any of the cities that were the biggest on population 500 years ago are still in that category. And then sort of in real time, as it's marching through, you can see sort of it's almost like a horse race to see cities grow and to decline. So like Istanbul, Turkey was the center of the most human population in a city as we're getting through the late 1600s. And then it's just right around 1700 where all of a sudden Tokyo comes and overtakes it. And then Beijing springs into the lead. It's really kind of fun to just watch sort of these population changes in kind of graphically illustrated in real time. So that was just kind of a fun graphic to consider human dynamics, not as measured by not as measured by genomes, but as measured by just people.
At 2005, so Tokyo is being overtaken by Delhi. It looks like. So currently Tokyo is the winner, right?
Yeah. So I'm still back in the 1840s now, and London is going crazy. Paris is right behind it. New York's coming on strong. In third place at the end of the 1800s. Pops into second place right around the turn of the century to 1900. Kind of interesting to think about.
So Tokyo is quite far ahead of everyone. 38 million, basically, yeah. And then Delhi is 27, and all the other cities are pretty similar. Shanghai, Beijing, even New York, 18 million,
Yep.
So the funny thing is, I was in Sweden. The population of Sweden is 10 million, less than the population of New York.
That's right. Yep. No, New York.
Denmark is 3 million.
Yeah, that's right. And here it's basically entire countries and cities. Yeah. And I think around the 1950s, New York had a brief moment at number 1. It's now number 10, at least as of 2018. So actually shrinking in the last 50 years pretty significantly relative to other cities.
Yeah, well, I mean, Tokyo will probably be overtaken by Delhi, I would guess. Mm-hmm. Eventually, in Shanghai, maybe Beijing. They're just crushing it there.
Yeah, coming on strong. That's right.
That's very cool. Very cool.
Kind of a fun graphic. How about you, Vincent? What's your science pick of the week?
Well, I have, before I take a pick, let me give you a link to this beer stuff, the history I was telling you about.
Oh, yeah. Is this the dogfish?
Yeah, so I was searching it in the beginning, and so it turns out that Patrick McGovern, he's at the University of Pennsylvania Museum of Archaeology and Anthropology. He's kind of a microbiology of beer historian. And he was involved in, I believe, a search for old beer. And they found pottery jars in a Neolithic village in Jiahu, which is in China, northern China, and they found 9,000-year-old mixture of fermented beverage, which is composed of rice, honey, and fruit. So they analyzed it by mass spec, and they kind of back engineered it.
Oh wow!
They hacked it, and they made a brew called Chateau Jiahu, and that's what I bought and drank. And so I can't make it a pick because I didn't really like it, but I'll put the link in the show notes for anyone that wants it. So my pick is a interesting story where they got a 1.7 million year old rhino tooth, and they got some protein sequence from the enamel.
Oh yeah, I think I saw. So I didn't read that, but yeah.
So tooth enamel is a protein. It's very stable. And, teeth can last a long time. So the story here, which is, I have a link in the Smithsonian article, and I'll also put a link to the original Nature paper. DNA sequencing can go back a while, and we've talked about Svante Pääbo's work. But the oldest DNA sequence is actually a horse DNA, 700,000-year-old permafrost frozen horse. So that's even older than Neanderthals, of course, because it was frozen. But you can get even older with protein. And so this thing is 1.77 million year old Eurasian rhinoceros species, Stephan orinus. They sequenced the bloody protein.
Wow.
Now,
that's pretty wild.
And they found, and this is interesting because it turns out that it's not an ancestor of the better known woolly rhino, which survived until about 100,000 years ago, 10,000 years ago, but it's a sister lineage. Now, the only issue here, of course, Nels, is that we're not going to be able to see positive selection because we don't have the codons, right?
That's right. Yeah. It's just the amino acid sequence.
The amino acid sequence. So it's kind of cool, but it's not going to tell you the whole story, right? But anyway, it's a cool thing.
Yeah, very cool. Really wild to think. I mean, it's just really fun to kind of daydream about what might be eventually possible as more and more stuff is sampled. Yeah. And as technology gets better as well. And will there actually be comparative possibilities? I don't know. I mean, there's obviously there's some physical limitations, right? In the stability of DNA relative to peptides, et cetera. But I mean,
this is teeth, so I don't know what else would be stable as well. I mean, they say that enamel is the most stable known.
That's protein,
right. But
exactly. Yeah.
You're going to be limited with teeth, but it's still pretty old.
Yeah, exactly.
And you can imagine if you could go back even further. I mean, the dinosaur. There's no dinosaur teeth, I guess. It's just fossilized bones, right?
Fossilized, yeah. So you're looking at rock.
Rock, yeah.
Yep. I just actually made a visit to Dinosaur National Monument. It's about a 4-hour drive. Was on the way, did a weekend at Boulder, Colorado. So former postdoc from my lab, Ed Chung, who is co-advised with Cedric Fichot, and actually was on TWiEVO, I think, with some of his work. He's now running his lab at Boulder. And anyway, he got married a couple weekends ago, but ended up stopping at Dinosaur National Monument and checking out this really great exhibit. So they built a building around basically one of the excavation sites, the Carnegie Quarry, And so there's now, dinosaurs at the Smithsonian, other museums across the country. But they said, and I'm forgetting the name of the guy at the time who started running it, said, let's leave those in place and put a museum around this. And it was really spectacular to see those fossilized dinosaurs just sort of in situ. It's just kind of a jumbled mess. But it was, but really, 1,500 bones, like a real hot spot of kind of half excavated. So they just like articulated half of them, but left them in the rock. Really fun to see that in that form.
That's
different
cool.
than what. Yeah.
Good.
Yeah.
All right. That's TWiEVO number 47. You can find it at microbe.tv/TWiEVO, where we have all the show notes and links to things we talk about. Of course, you probably listen on a player of some kind. If you do, please subscribe. We'd like you to do that so you get every episode. It's free, of course, but we know how many people are subscribing, which we'd like to know. And if you really like what we do, consider supporting us financially, help with our expenses. Go to microbe.tv/contribute. We have ways you can use Patreon or PayPal to give us as little as $1 a month. That's not very much, and it would really help us out if you all did that. We'll have other ways coming up in the future as well, but we'd appreciate your support. Of course, if you don't want to, that's fine also. We'll keep doing it.
Absolutely. Still having fun.
And if you have any questions or comments, TWiEVO at microbe.tv. You can find Nels, Elde at cellvolution.org, and over on Twitter, he's L Earlybird. Thanks, Nels.
Yeah, thank you, Vincent. And excited. We're coming up on 4 years of TWiEVO, and then we'll also have, I think, a fun in vivo episode in Germany. So stay tuned for some fun events ahead.
Be cool.
Yeah.
I'm Vincent Racaniello. You can find me at virology.ws. Music on TWiEVO is by Trampled by Turtles. You can find them at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, be curious.